Hello, good afternoon, everyone. Hope you guys had a good week so far. Let's get started with lecture 3. So this is this week's topic about cells. And let's start with the 4 basic principles of the cell theory. Okay, so the first part of cell theory states that all living organisms are made up of 1 or more cells. Okay. So if we think about like a bacterium, that's 1 cell. Okay. We think about humans. Oh, sorry, I have to plug in my computer. It's gonna run out of battery soon. Oh, just in the nick of time. Okay, if we think about humans, then we have like trillions of cells. Okay. So whether you are as small as a bacteria or as big as a human, we are all made up of cells, 1 or more cells. That is the first part of the cell theory. What's another part of cell theory? Anybody want to help me out here?
A cell is a living or smallest unit of life.
Yes, exactly right. So to paraphrase what you said, a cell is the smallest unit of life. Okay. Individually alive. As part of the multicellular organism. Okay. Joel has another one. Cell contains hereditary material. And that can be passed on. Okay, so it could be in the form of DNA, sometimes in the form of RNA. And the last one, Giovanna said they divide. That's right. So all cells come from pre-existing cells. Okay, so this is what we call cell division, right? Or mitosis. Okay.
Then,
yes, go
then
ahead.
where is the initial cells come from? If other
Yeah,
cells?
that is a very very good question, right? Nobody knows. If you believe in creationism, right, then perhaps God created the first cell. If you believed in evolution, then perhaps the chemicals, the macromolecules randomly came together to form the first primitive cell. But the truth is, nobody knows. But yeah, good question. Moving on. Okay. Fill in the blanks with terms that correctly describe the organization of life from smallest to largest, starting with atoms. Okay, so we learned last week, I think, atoms are the smallest unit of an element, right? And they could, of course, come together to form molecules. Okay, so an example of a molecule would be like H2O, right? Something like CO2. These are examples of molecules. And those are small molecules, right? Couple of atoms in the molecules. And we know that they could actually be much bigger, right? They could be macro molecule. Okay, so such as lipids, nucleic acid, proteins, and carbohydrates. Okay, and these macromolecules will be the basis, the basic building blocks for which we can use to make bigger things like organelles. Okay, so organelles are like these tiny structures that are found within a cell, And so the next level up from organelle would be a cell. Okay. So different cell types can then come together to form tissue. Okay. Does anybody have any example of tissues for me? Example of tissues?
Fat tissues.
Yeah, fat tissue, right? Fat tissue, adipose tissue, we call it. Anybody else have another one? Any other tissue type that you know of?
Just guessing, like a blood clotting.
Yeah, blood is a type of tissue. Yeah, blood is a type of tissue. Very good.
What
not
about
necessarily,
clotting? Like
not necessarily a clot though. The clot is when we talk about tissue, we talk about the kind of tissue that makes up like a normal organism, right? So clots is it's actually proteins, if anything. But good try. Blood is. And then, yeah, muscle. Leah said muscle. Right. So all these are example of tissue. There's connective tissue. There's nervous tissue. There's a bunch of tissues that we will learn later on in the course. So these tissues, different types of tissues, can come together to form an organ. Okay, and then multiple organs would form an organ system. Any examples of organ system? Anyone know of? Give me one example of an organ system.
like respiratory.
Yeah.
Urinary system.
Urinary system, sure. Urinary system. Cardiovascular system, someone else said, very good. Give me some organs that is in the urinary system. Anyone? Yes, Adriana, I saw you on mute. No? Yeah, go ahead.
Kidney.
Kidney? Yeah, kidney is in the urinary system for sure. The urinary bladder.
Urinary tract.
Yeah, so those are all examples of organs, okay? Heart is another organ, yeah, but that's not in the urinary system. That would be in the cardiovascular system, okay? Very good. So that's the organization. Moving on, list 3 common structures found in all cells. So even though cells are kind of, they look different, right? They might have special features that are specific to that particular type of cells. There are 3 things that are common for all cells, okay? So they all have a cell membrane. Another word for cell membrane is plasma membrane. Anybody remember what the major component of cell membrane is? We learned that last class, I believe. What's a major component of cell membrane?
Like they block something go in and they allowed something go out.
Right, that's the function. Yeah, very good. But what's the major component? Did I say function? I mean component. Major component of a cell membrane. What makes up the cell membrane? Anybody remember? That's right. Yeah, so a couple of people say yes. Skina said phospholipids in the chat. That is correct. Another function, another common structure found in all cells. Shivana said nucleus. Not necessarily, right? Some cells don't have nucleus. Joel says cytoplasm. Yeah, very good. Cytoplasm. So if you think about like a cell, right, that's the membrane that surrounds it. And then the fluid inside, the gel-like fluid inside, that would be the cytoplasm. Okay. And Vino said DNA. Yeah, some kind of genetic material. We don't necessarily know if it's DNA. It could be RNA, but there's some kind of genetic material. Okay. What are the examples of cell without a nucleus? Yeah, like a bacteria, right? Bacteria don't have a nucleus, right? Okay. So that's those are the 3 things that are found in all cells. Okay. Next one. Fill in a chart to compare prokaryotic cells and eukaryotic cells. So prokaryotic cells are primitive cells. You can remember P stands for primitive. Another name for prokaryotic cells is eukaryotes. Yeah? And another name for eukaryotic cells would be eukaryotes. Okay, so the prokaryotes, they are quite small. So the size is smaller anyways, compared to the eukaryotic cells. Because they are quite primitive, the structures are going to be simple. It's not going to be a lot of stuff going on inside. Very simple. And the location of the genetic material is usually located in the center. Okay, central area of the cell. Okay, so if you think about a bacterium, okay, bacterium, that is going to have some genetic material inside. Okay, so it's just going to be in this general area in the center. Okay, so those are the genetic material. Okay, now in addition to these genetic material that are in the center, the bacterium also have these circular DNA. Okay, does anybody know what these circular DNAs are called? Starts with a P. Anybody learn about them before or read about it? Starts with a P.
Prokaryotic cell or something like this.
Good try. Good try. Plasmid. Anybody heard of plasmid? No. We'll learn more about it later in lecture 9. Okay. How about this? Anybody heard of like superbug? Superbug. It's on the news all the time. Superbug. Right. Superbugs are these bacteria that have resistance to a lot of antibiotics, and so there's no way of not a lot of medicine that could treat them if you get infected by one of these superbugs. And what makes them super? What gives them the ability to be immune to a lot of antibiotics are these plasmid. Okay, so we'll learn more about that in, like I said, lecture 9. But yeah, that's where the genetic materials are. It's going to be sometimes in the center, and then you might have a few circular DNA kind of floating around in the periphery. Number of organelles, few. Okay, an example of prokaryotic cells would be bacteria. All right, anybody know any bacteria name? Just, from the news, from the radio, or you remember that from before? Yeah, usually this is the most common one that students give, E. coli, right? And so E stands for something really long that nobody knows how to pronounce. So we just call it E. coli. Yep, Salmonella is another common one. You always hear on the news that, whatever brand is recalling their salad because of Salmonella contamination, right? Okay, so these are examples of bacteria. Okay, they are prokaryotic cells. They are small, simple. Genetic material is not in the nucleus. There's no nucleus. In fact, only a few organelles. But don't be deceived by their simplicity. Okay, they could pack a punch when they infect you, right? Could make you very sick. People could die from E. coli and Salmonella infection. Okay. Yeah, someone else says Staphylococcus. Definitely, Staphylococcus is things that could cause like strep throat, or it could cause like a simple pimple on your body. Okay, fungus is not prokaryotic. Fungus is eukaryotic, but good try though. Moving on. Next, we have eukaryotic cells. Size are bigger. The structures are complex. And the genetic material is inside the nucleus. Okay. So like if you think about the cell here, there's going to be a centralized structure in the middle called the nucleus. And inside, that's where you will find your genetic material, your DNA. Okay. There are going to be many organelles. An example animal cells, plant cells, fungus, protists. Okay, I'm not sure if people know what protists are. Protists are like single cell organisms that lives in pond water, rivers, right? An example of protist would be something like an amoeba. Okay, you might have heard of that before. I do want to put a little star here, right? There are some exceptions, okay? In the human body, some cells don't have nucleus. Okay, even though we're supposed to be eukaryotic cells, some of our cells actually don't have nucleus. An example of that would be like RBC, white blood cells, platelets, right? The cells that helps you clot the blood, they don't have nucleus. Pseudomonas. Yeah, I think that's a bacteria, right? Pseudomonas, right? It's a type of a class of bacterium. Someone type that in the chat. So there you have it. Difference between prokaryotic cells and eukaryotic cells. Any questions before we move on? Okay. Please try this poll question. Someone keep on ringing the bell. Okay, I just gotta see who it is. I'll be right back. Okay, couple more seconds. Yeah, most of you got it right. The answer is, of course, D. A cell is a basic structural and functional unit of living things, but it's the smallest unit of life, right? That's what you have to remember. Very good. Let's try one more. Go ahead. Okay. We just talked about this though, right? Like, if you look at what we filled in, all cells are going to have cell membrane, cytoplasm, genetic material, right? So they are not necessarily going to have a nucleus. Okay? Not necessarily going to have a nucleus. In fact, all prokaryotic cells don't have a nucleus, right? Okay, so the answer is C. All right, moving on. Here we have a mitochondrion. Singular is RION, plural is RIA. All right, so let's go ahead and label the key structure of the mitochondrion. Of course, the function is to produce energy, right? Produce ATP using cellular respiration. And we're gonna learn about how they do it in lecture number 5. But for now, let's label some of the key structures. Okay, so here we have a outer membrane, okay, right here. This is cut open, right? Okay, it's like the actual thing is more like, if you think about like a jelly bean, right? That would be kind of like what it looks like. And now you cut it open and you look at inside. That's what the inside structures will look like. So here we have the outer membrane. outer membrane. And we also have the inner membrane that folds up into these accordion shape looking structure. They are called cristae. Okay, and by folding up like this, it is going to increase the surface area available for reaction to occur. So this is inner membrane, right? Forms finger-like, that's what it's called usually in the textbook, finger-like projection. Finger-like, they were like fingers. Projections called cristae to increase surface area, right, for chemical reaction. Okay, and these chemical reactions are the ones that are going to be making ATP, the energy, form of energy that we use. Okay. So these arrows are a little bit unclear, so I'll just redraw it here. You have your outer membrane, you have your inner membrane. It's a double membrane structure. Okay, so the space here, the space in between the outer and the inner membrane, that's what this is pointing at. It's pointing at the space between the two membranes. That is called the intermembrane space. Okay, we will learn about the significance of these areas later on when we do lecture 5. And then on the inside, right, inside of the inner membrane, this area, right, that area is the matrix. Okay, a lot of stuff is going on in the matrix. You're going to be making a lot of ATP from within the matrix as well. Okay, so these are the various structures of the mitochondria. Okay, fun fact, mitochondria have its own DNA, okay, which encodes some of the mitochondrial proteins. Okay, so where do we get the mitochondrial DNA from? Let's do a poll. Do you think we got it from A, mom, B, dad, C, both parents? Okay, who do we inherit the mitochondrial DNA from? Did I tell you this in the recording? I honestly don't remember. I recorded at the beginning of the pandemic, and I never watched my own recordings. Okay, interesting. A lot more people participating now. Very good. Yes, many of you say it is from both parents, but surprisingly, it's not. All your mitochondrial DNA comes from the mother. Okay, and this is why. Okay, where do we come from? Okay, my daughter actually asked me that question a few weeks ago. She's 7 years old. She's like, so where do we come from? Like, where do I come from? She said. I said it's you know, mommy has some DNA and daddy has some DNA, and you put it together, and then you get you. And then she's like, so like the DNA makes the hand and the feet, and they know where everything goes. And I'm like, you will learn it later when you grow up. But anyways, if you think about where we came from, it's actually like the egg, right? The egg, and then the sperm, right? Okay. So the sperm is going to inject the DNA into the egg, and then the egg is the one that has all the mitochondria and all the other nucleus, right? All the other cellular structure, right? So after the DNA gets injected into the egg, none of the structures from the sperm is going to become part of the zygote, right? Zygote is what becomes the baby. And then so everything else inside the egg is going to continue to grow to become part of the baby. So all these mitochondria that were there in the egg in the first place, those will become all of the baby's mitochondria. Okay, so in that way, everybody's mitochondria is exclusively containing the maternal DNA, okay, which is kind of cool. Moving on. Okay, now we are going to film this chart, and this is going to have everything about the endomembrane system. Endomembrane system. Okay, so what is the endomembrane system? So endomembrane system is basically a series of membranes inside the cell. Okay, and they have interrelated functions. All right. So what I'm going to try to do is I will summarize, like I'll do a little bit of description of the organelle, and then I will explain what it does. Okay. On the next quiz, there might be some diagrams. So I might show you a picture of this and I point to something and you have to tell me what the name is. Okay. It is going to be like you will have to type the answer in. Okay, let me explain a little bit here. So like if I point to this thing, right, and then I ask you to key in the name, and so this is of course the rough endoplasmic reticulum. Okay, which is also RER, right? Okay, rough ER. So when you type in the answer, please type in the full name and make sure you spell it correctly. Okay, if you spell it wrong, then the computer is gonna mark you wrong. Okay, I'm gonna go through it manually too, but then just to save it, save me some time, please type it in correctly and don't answer it in full sentence. Okay, don't say this organelle is rough endoplasmic reticulum. Just type the name. Okay, otherwise it's gonna throw off the computer, and it's gonna mark you wrong. Okay. But like I said, I'm going to go through it quickly as well, just to make sure that it's going to be okay. But just to give you a heads up. Do you guys understand what I'm saying? Okay. Like last time I gave a picture of the mitochondria and then someone typed the mighty mitochondria and then the computer marks him wrong. Okay. Like no need to add adjectives. Just tell me what the name of the organelle is. Okay. So over here, we're going to do some description and we're going to have some function. Okay, I'm going to do the description in pink and then function, I'm going to do it in blue. All right. So here we go. This is the rough endoplasmic reticulum. So if you spell it wrong, is it going to be wrong? Yeah, I mean, I'm going to go through it as well. If it's like 1 or 2 letter wrong, it's okay. I'm going to give it to you. Okay. But then like if it's way off, then it's not going to be good, right? I mean, it shouldn't be hard. You guys going to have your sheets in front of you, right? So you shouldn't be spelling it wrong, I think. So description, what is this? It is, of course, part of the endomembrane system, and it is continuous with the nuclear envelope. All right. So there is the nucleus. Don't draw this. I'm just showing you. Okay, there is the nucleus, right, which is what you will have in all the eukaryotic cells, with some exceptions. And then surrounding the nucleus is going to be the nuclear envelope. Okay, the nuclear envelope. Now watch. The nuclear envelope will continue to extend outward into the cytoplasm. Okay. And that's what will form the endoplasmic reticulum. Okay. Some of the endoplasmic reticulum will also have ribosomes on them, which gives them a rough appearance, and that's why it's rough endoplasmic reticulum. So that's what it means by continuous with the with the nuclear envelope. Okay, it's actually stuck to it, if you would. Okay, it's an extension of the nuclear envelope into the cytoplasm. Okay, so that's what it is. You know what? I kind of like this picture. I am going to keep it. I will move it up here. Okay, so just in case you forgot what I mean, this is the nucleus. Okay, the green thing here, that's the nuclear envelope. Okay, so continuous with the nuclear envelope, and it is covered with ribosome. Okay, which are all these like little dots there. Okay. So what does it do? What does the rough ER do? Give me a second. The rough ER is going to work with the ribosome, and they will package the proteins. Okay, so the ribosomes will synthesize proteins directly into the inside, the lumen, right? The inside of the endoplasmic reticulum. Okay. The ER will package the protein into a vesicle, which will butts off from the ER. Okay. So this is what it looks like in a low-tech animation. Okay, just try to visualize this. This is the ER. You're gonna have the ribosome right here on top. Oops, ribosome right here. And then the ribosome is gonna be making a protein. Okay, so there is your polypeptide. And keep watching. And polypeptide will fold up into a 3D shape. Okay, and then you're gonna take this and you're gonna pinch this off into a bubble, into a vesicle. And then you're gonna have the protein inside. Okay, so that's what this description means. The ribosome, which is the little pink dot here, will create a protein directly into the inside of the ER, shown here in black. This protein will fold up and move to the edge of the ER, where it will pinch off, where it will butt off into a vesicle. Okay, it's much better if you watch this on YouTube. And I think in one of the video resources, there is a nice video. It's called The Tour of a Cell, I believe, that shows you all these things. Are we together? Any question before I move on? Everybody happy?
I didn't get it. I don't know.
Oh,
I feel
part,
little fast. I didn't get it.
This last part you don't understand?
Yeah, it's a description here.
Okay, sure. Let me try that one more time.
Thank you.
Yeah, no problem. Okay, do you understand this is the endoplasmic reticulum? It's just like a bunch of networks, right? Okay, this is the endoplasmic reticulum. Okay, so the inside is hollow. Okay, it's empty on the inside. Okay, hollow like this on the inside. There's nothing. Okay, so now we're gonna have a ribosome again. Okay, it's covered with ribosome. Right? That's a ribosome, 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 ribosome. Is that okay so far? Anything that needs to be clarified from this picture at this moment?
Got it so far.
Okay.
Thank you.
Now, so the ribosome is going to make the protein, right? The protein is going to go inside, protein is going to go inside, protein is going to go inside. Okay. So this blue thing is the polypeptide, right? Polypeptide. Okay, which will fold up into the 3D shape into the protein, right? So there is the protein, there's the protein, protein, Okay, so these proteins need to come off from the ER. So it's going to move to the edge. Move to the edge, and then you basically take this part and then you pinch it off. Okay, so when you pinch it off, right, you're going to get a bubble with the protein inside. That's basically what happens. Okay. Hopefully that helps a little bit. And then again, watch the animation. It will definitely clear things up. I'm not a big fan of showing animations during these live sessions because I think it's better if you watch it on your own. Okay, moving on. So further out, right, you would have more ER, but these ones are free of ribosomes. There's no ribosomes on them. Okay, and therefore, under the microscope, they would appear to be smooth. And so we call them smooth endoplasmic reticulum, okay, or SER. Okay, SER. So let's go on with some description. It is continuous. Continuous with the rough ER. Okay, and it is not covered. Not covered with ribosome. Okay. Now, what is the function of this smooth ER? Okay, the function is whereas the rough ER works with the ribosome to make the proteins, the smooth ER is responsible for the synthesis of lipids. Okay, so what kind of lipids do we have to make? Well, it might be steroid hormones, right? Steroid hormones. Okay, it is also involved in the breakdown of lipids. Not only can you make them, you can also break them down as well. Okay, so everything lipids is going to be happening in the smooth ER. All right. So anybody remember any steroid hormone from last class?
Testosterone.
Yeah, testosterone, right? Yeah, cholesterol is the starting material of testosterone. It is not a steroid hormone itself. So testosterone, estrogen, right? So what kind of cells would you expect to have a lot of smooth ER? Anybody? Where do testosterone, progesterone, estrogen come from anyways? Well, which part of the body makes that? What structure, what organ makes that? Let's talk about the estrogen and progesterone first, okay? What structure makes estrogen and progesterone? That's right. The ovaries, right? The ovaries makes estrogen and progesterone, and the testicles makes testosterone. So in those structures, the cells of those structures, you would expect to find a lot of smooth ER. Okay. Moving on, we have the next structure here. Anybody know what that is? This next thing, if I show you this picture on the test, what are you going to type?
Golgi.
That's right. You're going to type Golgi apparatus, right? Or Golgi body. Both would be okay.
In my language, Golgi means like pocket. That is like pocket. That's I remember.
Pocket, yeah. It's like little pockets so you can put things in, I guess. Apparatus. Okay. Or Golgi body. Okay. Whichever one you prefer. So what is it and what does it do? Well, let's go with the description. It is going to be layers of membranes stacked together, right? not necessarily touching each other, but like really close to each other. And the function of the Goji is to sort, modify. It's almost like a slogan: sort and modify and repackage contents of vesicles. Okay. So let me try to explain this to you with my low-tech animation. Here we go. There is a Goji apparatus here with a few stacks. Okay, so what happens is that vesicle that buds off from the rough ER, it can come over here and then it will fuse with the first layer. Okay, and then you might modify it a little bit, add some stuff to it, and then a new vesicle will come off. And then this new vesicle will go to the next layer, okay, and then gets modified, and then it will go to the next layer, and sometimes it actually goes back. Okay, it's very organized. One side go forward, the other side go backwards. So there's kind of like this back and forth shifting until it is finally ready. Then you would have a final new vesicle. And the content here is going to be, modified. So maybe it has some added stuff to it or something like that. Okay, so think of this as an assembly line. Okay, so like the raw material comes, you're gonna add some things to it, you're gonna pass on to the next person, right? You're gonna pass it to the next membrane, and then they receive it. They might add some things to it, and then they look at it and like, oh, maybe you missed something. They will shift it back, and then back and forth until it is finally done. Then you get a final vesicle. Okay, so it sorts it, it modifies it, and then it repackages it into a vesicle. So this vesicle can be destined for a few destinations. It can go to a few different places. Okay, and you should know what they are. Anybody remember where can this vesicle go? Where will it go? Where is gonna go? Anybody?
Like different part of body where they need to go.
Different part of the what? I'm sorry, I just didn't hear you. Different part of the body. It could be, but let's talk about the cell first, okay? In relation to the cell, where will this vesicle go? Anyone else? Yeah. So Adriana said cytoplasm. It could certainly remain in the cytoplasm, right? For later use. Okay. What's another one? What's another possible destination? Cell membrane. That's right. It could embed the protein into the cell membrane. Okay. So next week you will learn about the structure of cell membranes. There's so many things on it. One of the things you can find on it would be proteins. Okay, and this would send the protein to the membrane. You can also release the contents, the proteins, out of the cell. Okay, maybe it will go into your bloodstream. Okay, and then it will get distributed throughout the body. And another possibility is that it will become a lysosome, right? Which is the next thing that is on our table. Okay, so that's the possible destination for our vesicle. Okay, I'll ask you a question about that in a second to see if you are paying attention. Okay, so here we go. The next part of the endomembrane system is the lysosome. Lysosome. Okay, what is the lysosome in terms of description? It is just a vesicle containing digestive enzymes. Okay, and it has various functions. It could digest macromolecules or ingested particles And sometimes your cells would eat things up. Like if you think about like a white cells, they would eat up a bacteria and then they need to digest it. So the bacteria, the lysosome will go and help with that task. It's also going to be involved in apoptosis, okay, which is programmed cell death. Okay, sometimes we want a cell to die because either it's like malfunctioning, it's too old, right? It's not doing its job properly. Then we want to get rid of the cell, right? For the benefit of the entire organism. Sometimes it's necessary to get rid of a cell, right? I think in the lecture I mentioned about during embryonic development, our fingers have webs in between them, right? So you want the webs to go away. So those cells need to die, and that is apoptosis. So lysosome is going to play a part in that. Right? It's going to release its digestive enzymes, and it will help break down those cells. Okay. Now, I mentioned a few diseases in the lecture related to lysosome. So these enzymes are, of course, proteins. And proteins, the instructions to make those proteins are encoded within your genetic material, right? So if there is some kind of mutation in the genetic material, it's going to cause your enzymes to malfunction, or you're going to make the wrong enzymes or something like that. Okay. So when that happens in one of these enzymes, we could have some lysosomal diseases, right? Okay. Anybody remember any lysosomal disease that I mentioned in class in the lecture recording? Anyone at all?
Tay-Sachs disease.
Sorry, the mic is really soft. I couldn't hear. One more time, please.
It's Tay-Sachs disease.
Yes, Tay-Sachs disease. That's right. Okay, very good. Tay-Sachs disease. Right. So in this one, we have an accumulation of lipids in the brain, right, in brain cells, which would then kill them and cause problems. This one can be screened using prenatal genetic screening. Rueda said Pompe disease. That's correct. This one is an accumulation of glycogen, right? Glycogen in muscle, right? Especially the heart. Okay, so depending which enzyme gets the mutation, you know, the sort of things that gets accumulated, it's going to be different. And depending on the location of the accumulation, the manifestation of the disease will also be different. So there you have it, the endomembrane system. Does anybody have any question before we do some more polls? Don't be shy. If you have a question, please do ask. Excellent. Okay, let's do the next one, which is this one. Go ahead. Okay, a couple more seconds for anyone else who wants to log in their answer. Okay. So if you track the movement of proteins produced in the rough ER, it can end up in all of the following except, and so it's this picture that I drew for you, right? This diagram, it can go to all these places, right? So if you compare this with the responses, we'll see that this would be the exception. It's not going to go to the smooth ER, right? The function of a smooth ER is to make lipids and break down lipids. Okay, so the proteins that you make will not go there. Let's see. Okay, that's it for now. Let's move on. There is something I need to change for this question. I keep on forgetting to change it in the worksheet. List 3 organs or structures that are found in plant cells. Not, I will cross out unique. We'll say found in plant cells, but not animal cells. Okay. So what are some things that are found in plant cells and not animal cells? Sakina said cellulose. Cellulose is not really an organelle though. So yes, so chloroplast is a good one. Chloroplast, let me, chloroplast. Okay, there is the cell wall, which is made up of cellulose. And then there's also, yeah, the central vacuole. Okay, so I don't want to say unique because chloroplasts are found in other cells as well, just not animal cells, but you can find them in protists, for example. Cell wall, you will find them in fungus, for example. So they're not unique in plants, but comparing plants to animals, these are only found in plant cells and not animal cells. So what does the chloroplast do? The chloroplast contains chlorophyll, which allows photosynthesis to take place. Okay, photosynthesis is how they make food. Cell wall. Okay, what does the cell wall do? It protects, provides structural support, right? Prevents the cell from bursting. It's not going to pop if there's too much water. And the central vacuole, which takes up 70 to 80% of the entire cell's volume, it is going to store water and nutrients. Okay, can also store pigment like colors and even poison. Okay, if the plant is poisonous. Okay, so those are some structures, organelles that you will find in the plant, but not the animal. All right. Moving on. Okay, so these are additional structures. First one. That is the nucleus. Okay. So the nucleus is found in eukaryotic cells, eukaryotes. Okay. It is surrounded by the nuclear envelope. Okay, which is this like purple covering. And then it protects genetic material. And it also contains the nucleolus. Okay. The shape of nucleus and that of lysosome are almost the same. Yeah, in fact, like a lot of stuff are actually look very similar, right? Okay, but the thing is, like, if you look at the cell, the nucleus is always very big and in the center, and then the lysosome will be much smaller. Okay, but you're right. It's hard to tell like the difference between like a lysosome and say like a regular vesicle. And then we also have these things called peroxisome. They all look exactly the same. Okay, so if you just look at an unstained cell, everything is transparent. You won't be able to tell what's what necessarily. Okay, which is why we can dye the cells. Okay, we can dye them with different fluorescing components, for example, and it will highlight a particular structure that we're interested in. Okay, so yeah. Anyway, so that is that. Okay, so what is what's going on here, right? What is the function, and let's go through that. Sorry, just looking for a bright color. Okay, so here, this thing in the center, right? That's the nucleolus. Okay, so the nucleolus, which is part of the nucleus, it is responsible for synthesis of RNA. Synthesis of ribosome. Okay, synthesis of ribosomes. That's where the ribosomes came from. Okay, and then the genetic material is going to be found over here in this location. Okay, so if you think about the nucleolus, if you think about the nucleus as a peach, and the fuzzy peach, the skin of the fruit is going to be equivalent to the nuclear envelope. Okay, the meat of the fruit, that's where you will find the genetic material, and the seed in the center, that will be equivalent to the nucleolus. Okay. Now, on the nuclear envelope, you will see that there are all these little bumpy circles. Okay, like all these openings. Okay, those are the nuclear pores. And maybe I'll label that for you. These are the nuclear pores. That's how things go in and out of the nucleus. Okay, so things like RNA, it's going to come out of the nucleus through those pores. Okay. So let's talk a little bit about the genetic material in the nucleus. And so we all know the genetic material is DNA, but the DNA is too long, right? It's not going to be able to fit into the nucleus if you don't package it. So you're going to add a little bit of proteins. Okay, for those of you who are interested, the protein is called histone proteins. Okay, then it's going to take the DNA and then it's going to coil it up into this structure, like very condensed form. So this is what we call chromatin. Okay, and that's the stuff that you will find in the nucleus. Just before cell division, the chromatin is going to condense further. Okay, it's going to condense and coil up even more, and that's going to form what we are used to seeing, right? The typical picture of chromosome, right? Okay, so chromosome is essentially same as chromatin. It's DNA, but super coiled up. Okay, and the reason for that is when it's all coiled up like that, it's much easier to move it around rather than having a loose thread like that. Okay, so there is the nucleus. Does anybody have any question about the nucleus? Okay. It is 1:30, so why don't we take a 5-minute break and we will do more when we come back. Okay, if you have any question, please leave a message in the chat box and I'll get back to you afterwards. Okay, see you in 5. And we are back for more exciting Biology 1. Next part of the chart is the ribosomes. Okay, so they are very, very small. You can hardly see them on the diagram, but they're there, and they're very important. Let Let's write down their name, ribosomes. Okay, ribosome. They are found scattered throughout the cytoplasm. Okay, either existing freely on their own or attached to endoplasmic reticulum. Okay. Sorry about the messy writing. If you cannot read it, just let me know. I'll be happy to read it to you. Okay. And the function is protein synthesis. Okay. I should also make another note. It is made up of not having enough space here. Made up of RNA and protein. Okay. And the ribosome, if you zoom in, actually looks like this. There is a big subunit and then there's a small subunit. Okay, kind of like a hamburger, if you would. So there's a large subunit and then there's a small subunit. Okay, so together it makes up the ribosome. Now is an appropriate time to talk about the central dogma of biology. Okay, dogma means like central theme, right? If you would. Okay, so the flow of information in biology, in a living organism, goes like this. And you start with DNA, which is the instruction to make all the proteins, right, in your system. And we are going to have DNA inside the nucleus. But to make the proteins, we need to do it in the cytoplasm, right? Because that's where the ribosomes are going to be. And it's too precious of the DNA to take it out of the nucleus. It might get damaged and it's too big anyways. There's no way to take it out of the nucleus because of its size. So what's going to happen is you're going to make a copy of some of the DNA. You're not going to make all the proteins all at once, right? You're only going to make a specific protein at any given time. And so you will copy the instruction that for that particular protein that you need, okay, in the form of RNA. Okay, so think about like having this master cooking cookbook, right, in the library. Okay, there's so many recipes in it. Okay, and then you want to make a specific you want to make a cake or something. Okay, so you're not gonna photocopy the entire cookbook. You just go to the page that you need, photocopy what you need, and then take that out of the library, right, and take it home. So same thing. We're not gonna copy the entire DNA. We only copy what we need, and we copy it in the form of RNA. So this process of copying some of the DNA in the form of RNA, we call that transcription. Transcription. Okay, and this happens in the nucleus. Okay, once you make the RNA is going to exit through the nuclear pores, and it's going to go into the nucleus. It's going to go to the cytoplasm, rather, where it will be used to make proteins. Okay, so that is translation. Okay, so there is your ribosome, and then you're gonna have your messenger RNA, right? This is the messenger RNA, and then the ribosome will read the messenger RNA and use that to see what amino acid to link together, right? And so you're going to get your polypeptide. Does that make sense? Okay, so that's how they are all kind of related, right? So we make our own messenger RNA, right? And you probably know by now that the vaccines, right, the COVID vaccines, the Pfizer and the Moderna, they are called mRNA vaccine, right? So instead of we having our own mRNA, right, those mRNA in the vaccine, they code for the spike proteins on the SARS-CoV virus, right, the COVID virus, right? So when they inject the mRNA into our system, this mRNA is going to be picked up by one of our white blood cells. It's going to go in our white blood cells with the mRNA, and then they're going to use the instruction on the mRNA to make the same spike proteins that are found on the virus, on the COVID. Okay, and then your body will make antibodies for those spike proteins. Okay, so when the real virus comes, you will have the antibodies ready to fight them off. Okay, so that's essentially how mRNA vaccine works, right? In a very simple sense. Okay, back to here. We have the mitochondria once again. Okay, singular is ION, plural is IA, and the structure, right? You can look at the diagram from before. So see question 5 for structure, and with the outer membrane, inner membrane, so on and so forth. The function, however, we should write down the function. The function is energy production. Okay. It's the main site for cellular respiration. Okay. So cellular respiration is you take glucose and you react it with oxygen. Oxygen we breathe in to produce carbon dioxide, water, and ATP. Okay, so you might be learning about balancing equation in chemistry. So for each glucose that we metabolize, we can produce up to 36 ATP, okay, in this cellular respiration process. Okay, so that takes place in the mitochondria. Plant cells, they also have mitochondria. The difference is plant makes their own glucose. We have to eat our glucose, okay, from our food source, right? Okay, next one. Anybody can tell me what this is? What are these pasta looking things? Looks like one of those fancy pasta. This is the centriole. That's correct. Thank you. Centriole. Okay. So some description. They are exist as a pair. Okay. Not found in plant cells. Okay. And the function is to produce spindle fibers. That will guide chromosomes during mitosis or cell division. Okay, so something has to move the chromosome around. You know, you got to separate them. You got to move them to one side of the cell. All that is done by the spindle fibers. Okay, spindle fibers. All right, next on the list, we have the cilia. Cilia, they are hair-like structures that covers the cell. Not all cells, obviously, right? some cells, for the cells that have cilia, usually it will cover the entire cell. Okay, so what does it do? It would enable movement, right? And in some cases, move things around the cell. Okay. So where do we find, sorry, yeah, someone want to say something? I hear someone unmute. No? Okay, maybe it's an accidental unmute. Where do we find cilia? Well, remember I talked about those protists? Okay, those single cell organism that lives in a pond. There is a common one called a paramecium. Okay, you don't have to know the name, I'm just showing you. Okay, it has a mouth and then it has a pump. Okay, and then it's covered with cilia. Okay, you might remember seeing this guy in like high school biology. Okay, so these cilia, they kind of beat back and forth like that, and then it helps the cell move. Okay, what about in the human body? In the human body, where do we find cilia? The mesola, is that how you pronounce your name? I'm sorry if I am pronouncing it wrong. You said euglena, that's correct. But euglena is it has flagellum instead of a cilia. Okay, but I'll write down. It has 2 flagellum. For human body, where do we find cilia? Tell me 2 locations where you will find cilia. Anybody? Sperm does not have cilia. Sperm has flagellum. Anyone else? Esophagus? No cilia in esophagus. Esophagus is your food tube, right? You just squeeze the food down, okay? Like you are squeezing a tennis ball through a stocking. We don't need the cilia. Airway, that's right.
How about trachea?
Trachea, that's right. Okay, so
Yeah.
trachea. Okay, so this is your trachea, your windpipe, right? Okay, it has a lot of cilia inside. All right, so like if there is a bacteria that gets stuck there, okay, if you breathe in some dust particle, the cilia is going to sweep them up, right? And then you can either spit it out or you can swallow it. Doesn't really matter. The point is you can sweep them out. Okay, so trachea, that's one of them. Okay, not so much in the lungs, not so much in the nose, but mostly in the trachea and the bronchi. Okay, the trachea splits into 2 smaller airway. Okay, what about another one? Exclusively in female only. Right? Men don't have this. Which part? If you watch the lecture recording, you might remember me saying it. I must have mentioned something like that. No? Okay. Doesn't really look like it, but this is the ovary. Okay, and then the egg is going to come out, and then in the fallopian tube, right, which is the tube that leads from the ovary to the uterus, it's going to have these cilia that would guide the egg, right? Fallopian tube. So that's what it means by like move things around the cell. So there might be stuff that are around the cell. In the trachea, it would be like dust particles and mucus and stuff like that. In the fallopian tube, it would be the egg. Okay, so those are the location where you will find cilia. Okay, next we have the flagellum. Flagellum or flagella if you have 2. Sperm have only 1 tail. Okay, if you have 2 tails, the sperm doesn't know how to swim. It just go around in a circle. You can look it up on YouTube. Search up like sperm with 2 tails, and you will find interesting videos of sperm going around in a circle. So usually just 1 or 2 per cell, and it is responsible for movement. Okay, so that is all. Again, any question at all? You guys have been very quiet. I don't really know if you're with me or not. I hope you're with me. The quiz was okay. We have a class average of what was it, 68%. I would like to see a little bit higher, but it seems reasonably okay for the first quiz, I guess. Like I said, I won't be able to return the quiz to you, but if we have a little bit of time at the end, I'll go through some of the questions that a lot of people got wrong on, and we can, look at them. Okay, so let's add a few more things here. Cytoskeleton. Okay, so there are three major components of the cytoskeleton. The first one is the microfilament. Microfilament. Okay, so it is made up of. Again, all these things are in the notes, right? I'm just writing it out for you. Made up of actin, okay, which is a type of protein. Okay, it's gonna have like two actin strands, and they're gonna be like braid together, okay, intertwined together. What is the function of microfilament? It enables movement. Okay, so a cell doesn't need to have cilia or flagellum in order to move. Okay, you can think of the cell as like this sometimes it's like this blob that just kind of sits on the surface, and then if it wants to move, it can just extend the front, grab on, and then pull itself like this. Okay, again, it's very cheap illustration right there, but hopefully it gets the point across. It grabs onto something and then it pulls itself forwards, right? So in order for it to be able to do that, it relies on these microfilaments. Okay, so think of, again, like a blob here on a surface. Okay, and then if it want to move forwards, it literally would extend the front, and then it would grab on, and then what happens is the rest of the cell, like this part, would then go forward, and then that's how you inch forward a little bit at a time. So how does it do that? Well, it's going to rely on these microfilaments. Okay, so you will find a lot of microfilaments in muscle cells, for example. Okay, what's another example of a cytoskeleton that we talked about in class besides the microfilament? Anyone help me out here, please? Thank you. That is the intermediate filament. Okay, so these intermediate filament, what it does is it forms connections between cells. Okay, structural support. Okay, so if you think about two cells next to each other, and we don't want things to leak between them, and we have to put a lot of intermediate filaments there. Okay, it's going to staple them together, glue them together so nothing leaks through. Right, that would be particularly important in the intestinal tracts, for example, in blood vessels. We don't want things to leak out of them. Right. Okay, and the last one, Joel said microtubule. Thank you. Okay, so the microtubules, it forms things like cilia, flagellum, okay, spindle fibers. Okay, it also forms tracts for vesicle to move on. Okay, so in one of the videos I post, you can see the animation shows that there's a track, and then there is a vesicle, and then there is a molecule that looks like a tiny person, and it's like carrying the vesicle, and then it walks along the microtubule. So that actually is what happens in real life. Okay, the molecule that carries the vesicle actually does look like a person, and then it's going to use ATP to walk on the microtubules to take the vesicle to where it needs to go. Okay, so things are not randomly going on inside the cell. It has all these guiding systems. Okay, wonderful. Let's do another poll. There we go. This is a multi-select question. Go ahead. Okay, a couple more seconds for people to lock in their choices. Okay, majority of you got it right. It is protein and RNA. Okay, it was in the notes. We wrote it down. This is another one. Go ahead. Anyone else? Okay. The correct answer would be lysosome. Okay. The key word here is contains enzymes, right? There are a lot of different enzymes in the lysosome that have various functions. They break down different things, right? So that is the correct answer. Okay, here is another one. Again, please participate. Let's see, one way for me to tell if you guys are with me or not. The polls are anonymous. Okay, last call. Right, so let's go through them. First one, we're looking for the false one, okay, the one that are incorrect. Smooth ER is part of the endomembrane system. That's correct. Okay, the endomembrane system has the rough ER, smooth ER, and then we have the Golgi body as well as the lysosome, right? Muscle cells are expected to be abundant in mitochondria. That is also correct. Muscle cells You have to move your muscle. There's a lot of movement. In order to do that, you would need to have energy. And so, since there are a lot of movements, you would expect to have more mitochondria to meet the energy demand. There are multiple nuclei in a single brain cell because high processing power is needed. That is not true. Just one nucleus per cell. All cells have cell wall to protect the cell from the environment. Also not true. Right? Animal cells do not have cell wall. All right. So those are the false statements. Another one. The last one. Okay. The correct answer is microfilaments. Right? We wrote it down here. Microfilaments are made up of actin. Right? I said 2 long strands braided together, right? Intertwined together. All right. So those are some sample poll questions. Hopefully you found them helpful. Let's move on to the next part. We're gonna find the cell part that is responsible that match the description. Right. First one. Directly involved in protein synthesis. That would be the ribosomes. Okay. Again, protein synthesis, we call that translation. The region inside the cell, except for the nucleus, that would be the gel-like substance called cytoplasm. Next part, organelle that manages or controls all the cell functions in the eukaryotic cell. That sounds like nucleus. Okay, the nucleus contains the nucleolus, surrounded by nuclear envelope. Right? Digest excess or worn out cell parts, food particles, and invading virus or bacteria. That is lysosome. Contains a mixture of enzymes. Small bumps located on portions of the ribosome. Sorry, on the endoplasmic reticulum. The answer is ribosome. Involved in detoxifying harmful substances and digestion of fat, produces hydrogen peroxide as a byproduct. If you look at this, like it sounds like smooth ER, right? Digestion of fat and then detoxify harmful substance. You might think about lysosome, but then the key word here that distinguish the answer from all the stuff that we mentioned is the hydrogen peroxide. Okay, so there is something called peroxisome, right? So again, there are some overlapping functions between the organelles, but there are usually some key terms that separate them. So peroxisome uses oxygen to break down things, and it generates hydrogen peroxide as a byproduct. Produce usable form of energy in the cell, that is the mitochondria. Sorts and modified contents of vesicles, that would be the Golgi body. Site where ribosomes are produced, nucleolus, inside the nucleus. Provide support for cell, we'll just put cytoskeleton. Specifically, the microtubules can do that. The intermediate filaments can do that. Small hair-like structures used for movement, cilia. Forms a selectively permeable barrier around the cell. That would be the cell membrane or the plasma membrane. Long whip-like structures used for movement. That is flagellum. Contains chlorophyll, chloroplast, large sac filled with water, central vacuole, produces microtubule fibers needed for guiding chromosomes during cell division. That is the centrioles. Okay. I think the long answers 1, 2, 3 are pretty self-explanatory. If you need clarification, please do let me know. I will go through the application questions now. Okay, so let's take a look at the first application question. Speculate what will happen to an animal cell if it is placed in pure water. Do you expect the outcome to be different for a plant cell? Okay, so if I put in, let's say I have a beaker here and then it's water and then I put the animal cell in. Okay, so the animal cells has a lot of stuff in it, right? There are dissolved nutrients, some electrolytes perhaps. So what happens is the water will be drawn into the cell. Okay, so the animal cell will swell and potentially burst. So we'll talk more about this next class. But the plant cells will not burst. Okay, plant cells have a cell wall, right? They will not burst. Okay, that's what the cell wall is. Number 2 is related to what we were talking about earlier. Your heart muscle needs to function continuously through life. Which organelle would you expect to be most abundant in heart cells? Okay. Anybody want to tell me the answer to that? The mitochondria is correct. Very good. Mitochondria. Okay, because it produces ATP continuously, right? Okay, last one. Endocrine glands produce and secrete hormones. Which organelle would you expect to be most abundant in cells of an endocrine gland? Okay, this one's a little bit tricky. Anybody want to give that a try? What kind of organelles do you expect to have a lot of for an endocrine cell? Okay. Endocrine cells are cells that produce and secrete hormones. Vathani said thyroid. Thyroid is an endocrine gland, but I'm talking about what kind of organelles do you expect to find a lot of in those cells? So let's say in the thyroid gland, right? What kind of cells do you expect to have a lot of? Mora said smooth ER. Okay. Wada said the same thing. Yep. Perhaps, okay. It depends on what kind of hormones we're talking about. Remember, hormones can be protein hormones, such as insulin, right? Or it could be steroid hormones, right? Such as testosterone and stuff that we talked about, the sex hormones. Right? So if the hormone is steroid hormone, then you would expect to have a lot of smooth ER. If it's protein hormones, however, then you would expect to have a lot of rough ER, right? Ribosomes, maybe. Okay, and you would expect to maybe have a lot of Golgi body as well. Okay, so depending on which kind of hormones you're talking about. All right. So that's it for this week's lecture. Does anybody have any question? before I take up some of the questions from the previous quiz? Okay. Does anybody have any comments about the last quiz? Did you find it to be fair? Did you find it to be too hard? Not enough time? Just right? Any comments concerns you want to bring to me? Next quiz, do we have application questions? Yes. Next quiz, we will have application questions. I don't remember how many. I'll let you know. I will let you know when we get closer to the day. You guys still hung up on that breast milk thing from last week. I hope that's not the only thing you remember from last class. All right, if you don't feel comfortable talking about the quiz here in the public space, feel free to send me an email if you have any concerns. If you need extra help or anything like that, please do contact me, okay? All right, so let's take a look at some of the questions that many people got wrong. Okay? Again, you might not have these questions. It's all randomized. I'm not sure if you got these exact same questions, but these are the ones that are incorrect, answered incorrectly by many people. So here we go. First one. Which of the following is not true regarding macromolecules? Okay, so first one, cell use macromolecules to signal other cells. That is correct, right? That's what hormones are for, right? Hormones are proteins, hormones are lipids, and we can use them to signal other cells. So that is correct. Cell use certain macromolecules to store energy. That is also correct. We can store energy in terms of lipids, in the form of lipids, in the form of glycogen. Those are all macromolecules, so nothing wrong with that. An example of a macromolecule is protein. That's also correct. Cell produce macromolecules by the process of hydrolysis. You should remember hydrolysis is when you have a protein like that and you chop it up. Okay, not protein, but when you have a polymer rather. When you have a polymer and you want to chop it up, you have to do hydrolysis, right? Hydrolysis into individual monomers. So if you want to make macromolecules, you are not going to be doing hydrolysis. You'll be doing condensation reaction. So for that reason, this is not true. Okay, the next statement is also correct. This one, also poorly done. In certain species of animal, 33% of bases in its DNA are A. So if you have 33% A, what else are you also going to have 33% of? Which other nucleotide are you also going to have 33% of? Help me out here, please. C-test said T, and that would be correct because A always pairs with T. Okay, so if you have 33% A, you're going to have 33% T. If you add these things together, you will have a total of 66%. Okay, so what about the rest of them? Well, if you take 100% minus 36%, you will get 34%. Like 100 minus 66. 100 minus 66 gives you 34%. So the remaining 34% will be C and G, and they are going to be equally distributed. So if you take 34 divided by 2, that will be 17% cytosine and 17% G. And the question asks for C. So the answer is 17. Okay. Next one, in regard to lipids, the term unsaturated refers to, it is not talking about the rings, okay? It's not talking about the glycerol. The glycerol, it's common for saturated and unsaturated, right? The lack of double bond between adjacent carbon atoms, if it's unsaturated fat, it will have double bond, right? So it's not the absence of double bond, it's actually the presence of double bond. So that's incorrect. Butter, lard, those are saturated fat, not unsaturated. And then the last one, the presence of double bond. That is the correct one. Okay, next one. Disulfur bonds are often found to stabilize which of the following levels of protein structure. This one is tertiary. If you look at the PowerPoint slide, there is a picture that shows all the interactions between the side chains. It looks something like this. I mean, I don't want to pull it up right now, but if you go to the PowerPoint, there is a picture that shows a backbone of the protein. And then it will show like, you know, ionic interaction, okay, hydrophobic interaction. And then there's one that says like disulfur bridge, okay, which is basically when two sulfur bonds together. And that is part of the tertiary tertiary structure. Alex asked, did I do the wrong quiz? I don't remember any of these questions. Yeah, we all have different quizzes, right? Okay, so you might not get any of these questions, which is totally fine, right? The bank is like there are like hundreds of questions in it, so you might not get it. But these are the questions that appeared on a lot of people's quizzes, maybe, and then they are poorly answered. Okay, so yeah, if you don't see any of this, it's okay. And I don't know if I should say I'm sorry it didn't show up. Maybe you're glad you didn't get any of these questions. I don't know. Okay. In the human body, the enzyme lactase breaks down. So lactase is the enzyme that breaks down lactose, right? Okay, we just changed the ASD back to OSD to see what it does. And lactase breaks down lactose into glucose and galactose. Okay, that's the correct answer. All right. So yeah, thank you for coming. We do not have anything due next week. There's no, I don't think there's a quiz next week. Let me just double check. I think the next quiz is going to be in maybe 2 weeks time or something. Let me just double check. You guys are 201. Next week is February, right? Yeah, nothing next week. Next week is just going to be another lecture 4. And then Adriana said, the next quiz, do you mind helping us out with the application questions? Well, that's why I'm doing these things with you for, right? Like these are some of the application questions, right? That you should think about. I will maybe I'll do a few of, actually, you know what? Some of these poll questions are also going to be similar to the think application question. Like for example, this could be an application questions, right? Where you have to think a little bit. I'll see. Okay, maybe I'll pull up a few more things to practice on. But other than that, okay, have a great weekend, and I'll see you same time next week. Okay, bye for now.
Huh?